So historically, by conventional testing, one uses a large group of animals to be controls, no treatment, versus those that are treated to see if a drug has a statistically significant effect against tumor growth. Now, in the case of pediatric cancers, the reason we do this is because there are relatively few clinical trials that can be conducted on kids to test new drugs, in part because pediatric cancers are rare, and secondly, the 5-year overall survival or event-free survival is now approaching or exceeding 80%, certainly in the United States. So there's very few patients that are eligible for experimental drugs. So using preclinical models gives us the option of looking at a large number of drugs and to prioritize what goes further into the clinic. The reason we've taken the approach of single mouse is that one of the failures of conventional testing has been that because we're using the resources using a large number of animals, we could only look at a few tumor lines. So if, for example, one took Wilms tumor, a tumor of the kidney, we could only look at, say, 3 different Wilms tumors in mice. And that certainly doesn't adequately represent the genetic diversity of those tumors. So what we did was to analyze about 10 years of data where we had done conventional testing. And this was 83 xenograft models, that is, human tumors grown in immune-deficient mice, and 67 drugs, and asked a really simple question. If we had just used 1 mouse instead of 10 in the treatment group, would we have come up with the same answer? And the result was quite astounding. 80% of the time, it would have been absolutely correct. And if you allowed for a slight deviation of plus or minus 1 response category, which didn't make any difference to the outcome, the results were 95% concordance. So this allows us now to say, well, let's just use 1 mouse per tumor line. And instead of looking at 5 tumor lines, so let's look at 50 tumor lines of this particular cancer type. Now we can incorporate the diversity, the genetic and epigenetic diversity that we know exists within these pediatric cancers. As I said, we did a retrospective analysis of over 2,100 studies. And that gave us the confidence that these models were quite reproducible and the 1 mouse would work. So in the work we presented at the EORTC symposium, we did 5 prospective tests. 3 were done in leukemia models by Richard Locke's group in Australia, and 2 were done in solid tumors. And in the case of leukemias, we used between 70 and 90 different leukemias in mice. So each leukemia was from a different patient. And in the case of solid tumors, we used 34 and 50 different models from different patients to represent their diseases. And the concordance is about 95% again. So in a prospective test, it shows that this single mouse study identifies tumors that are very sensitive to these drugs and those that are essentially resistant to these drugs. And that allows you now to probe the molecular characteristics of these tumors, because all of these tumors are linked to RNA-seq and whole exome sequencing. So we can look for certain mutations that could confer sensitivity or resistance, or certain expression profiles that again relate to the drug sensitivity of a tumor model. Well, I think the results that I mentioned are really the key highlights, in the sense that as we go forward, and we run a very large program in United States to identify drugs that are really very active and should be prioritized to move into pediatric clinical trials, that we can now do this on a much larger scale, and perhaps over the next 5 years, not only identify drugs that are really very active, but they're very active in specific subsets of cancers, and be able to identify the characteristics of those cancers so that we can stratify patients that would ultimately benefit from these new drugs. I think in the United States, we just passed the so-called RACE Act, which is an act by which the FDA is asked to prepare a list of drugs in development and their targets that they interact with. And if that target is relevant to the growth of pediatric cancers, the FDA can mandate that that drug is moved forward into testing in pediatric cancers of the appropriate molecular type. Where I think the single mouse study is going to be advantageous is that we can select models that are driven by specific signaling pathways or by specific cancer genes, where these drugs are purportedly going to interact. And we can take those models and test the drugs to see whether these drugs are really effective in the molecular subtype that they're being developed to test. And the reason for this is that we know that certain mutations confer sensitivity to, say, signaling inhibitors, such as BRAF inhibitors, which are very active in melanoma. But we know that the same mutation in colon cancer does not confer sensitivity to these drugs. So even though you have a common genetic defect or a mutation that activates an oncogene, it's still, the response of the tumor is very context specific. So just because it works in an adult cancer does not mean to say that the same mutation in a pediatric cancer will confer the same level of sensitivity. So I think these are the sort of questions we can ask. And you know, if the drugs are active, is to move them quickly into the appropriate molecular subtype of pediatric cancer. I think this idea that we categorize tumors by their histology, by their site and such like, is still valuable. But as we go forward, we're going to be subtyping tumors at a molecular level, and hopefully our drugs will be specific for that molecular subtype and have very marked anti-cancer activity without the toxicity that we have from current cytotoxic therapies.